What I'd like to do today is talk about the carnivores, specifically not just animals that eat meat. Instead, I want to talk about the group Carnivora within the Mammalia. So the point is that carnivory and the carnivora are not synonymous. Most carnivores, that is, most carnivora, are specialized for eating meat, but certainly not all, and we'll um, see that when we begin to look at the teeth of these animals in a little bit. Many other non-carnivora consume meat, but those aren't the animals that we're concerned with now. When we think about the carnivores, we divide them up into two suborders. There are the ones that look like dogs or are very dog-like, and the others are very cat-like. And we refer to these as the caniniforms and the feliforms. One of the obvious differences between these two groups, one of several, but one obvious difference is that the caniforms have an allosphenoid canal and the feliforms do not. The allosphenoid canal is part of the carotid circulation. And here you can see an illustration of the allosphenoid canal. So anytime you find a carnivore skull, if you flip it over, and you can find that allosphenoid canal, you know that you're holding a caniform carnivore rather than a feliform carnivore. So then, if you look at a California sea lion, you'll notice that it does have this allosphenoid canal, which right away tells you that evolutionarily sea lions are more closely aligned with the dogs, with the caniforms, than they are with the cats. Here in this tiger, there is no allosphenoid canal, so it's obviously a feliform. There is another difference between feliforms and caniforms, and that's how the auditory bullae are constructed. In caniforms, the auditory bulla is just one single unit, and it's made up of the tympanic bone. In feliforms, however, there are two bones that comprise the auditory bullae. They are the tympanic bone and then the endotympanic bone, and you get the septum that forms between them. So as you look at the civet, you'll notice that it does have this sort of septum, which goes right down towards the anterior margin, but it divides the auditory bullae into two parts. So the civets are within the cat group rather than the dog group. Here, on the other hand, is a palm civet, and the palm civet does, in fact, have an allosphenoid canal. Now, if you look at a phylogenetic tree for the carnivora, you can see that we've divided them up into the caniforms and the feliforms, and then there are the creodonts, which form our outgroup. But notice what happens. So we showed that the civets, which are in the feliform, do have that allosphenoid canal, but that's sort of atypical. So clearly the presence of the allosphenoid canal isn't rock solid as a cladistic character, but it does give you this way of separating these two major groups. On the other hand, that endotympanic bone being involved in the formation of the auditory bullae is a good definitive character to divide these two groups. Now let's look at who the members of each group are. The feliforms include the felids, which are the cats, and interestingly, the hyenas as well, so the hyenidae. So hyenas, even though their heads look very dog-like and perhaps their bodies look dog-like, are not in the caniforms. Instead, they are cat-likes. And then there are viverids and the herpestids, which are the mongooses and the civets and all of those animals. So the smaller carnivores, those are all feliforms. Within the caniforms, you obviously have the canids, which are the dogs, the mustelids, which are the weasels and the minks, and the skunks, animals of that sort, the procyonids, which are the raccoons, Ilurus, which are the pandas, then the ursids, which are the bears. And then you'll notice you also have this group referred to as the pinnipeds, and those are the otobenids, which are the walruses, the phocids and the otariids. Those are the seals, the earless seals or the true seals, and then the eared seals. So there's another definitive character that we use when you look at the feet. The pinnipeds have these feather-shaped feet, and they're all aquatic. And then the fiscipeds are the split-footed carnivores, and those are all of the other caniform carnivores, so they have split feet. So it's here's a nice example, a gray wolf, a nice example of a caniform. No one's ever confused a wolf for anything other than a wolf. I suppose with the exception of some of the smaller subspecies, which are in the range of size of dogs, right, rather than the northern gray wolves. Gray wolves achieve a size of about 175 pounds and stand about 3 feet tall at the shoulder. Their heads are as big as basketballs, and they are pretty impressive predators. There is another wolf that, well, there are several other, one other species of wolf and one other subspecies of wolf in North America. That's the Mexican gray wolf, which is in the southwestern United States, and that, of course, is a species that is imperiled, and its situation hasn't improved with recent efforts to build a border wall between the U.S. and Mexico. The other wolf that we have in North America is the red wolf, which there was considerable controversy about whether the red wolf was actually a wolf or whether it was simply a hybrid between coyotes and wolves. But it turns out it is a distinct species. One of the reasons for that controversy relates to the size of the sagittal crest. And it turns out that the original descriptions of red wolves were all based on museum specimens, and all of the museum specimens came from zoos. And of course, wolves in a zoo setting do not get a normal sort of diet. They're fed dog food. And as a result, they're not using the temporalis muscles nearly to the extent that gray wolves are. And as a result, the sagittal crest is much less pronounced. Bone is dynamic tissue. It responds to the forces that are applied to it. And if you don't use it, then the bone is reabsorbed. So the original descriptions were for these, for red wolves, were for these animals that had minimal sagittal crest. And of course, that was sort of an artifact of the sample. Now we understand that they are a distinct species. One unfortunate aspect of the red wolves, and that's also something that's happened recently during the Trump administration, there are, I think, 3 extant feral populations of red wolves. All of them are supplemented genetically by zoo specimens, and there was a population viability analysis done by the U.S. Fish and Wildlife Service in which they meticulously established a plan for the long-term persistence of the red wolf populations, and it involved maintaining these zoo populations to supplement the genetics of the wild populations. And that entire program was shut down underneath the Trump administration. So the Trump administration removed all support for the red wolf populations, and as was indicated in this population viability analysis, without that sort of genetic support, those populations are doomed to extinction. So that's a rather unfortunate sequence of events. Here's a tiger. These are big cats. We'll talk a little bit later on about the different strategies that that cats use, or that carnivores use, to capture and subdue prey. These animals are quite impressive. One thing that I want to point out, if you look at the ears, you'll notice these white patches on the backs of the ears, and there's some debate about what those things are for. It's pretty characteristic. You see it in lots of felids. What is interesting is you can take newborn tiger kittens and you can present them with a piece of cardboard that has been painted black and has 2 white spots on it, and they will follow it. So one hypothesis is that these white spots are used as sort of a communication tool to indicate to the kittens that they should follow the female. So then we've made the distinction between the caniforms and the feliforms. and that's all pretty straightforward. We could think a little bit about mustelids, which are a little bit more complicated. There are two suborders, and we've noted that based on the structure of the auditory bullae. And also, and historically, we've used the characteristics of the feet, whether the feet are feather-footed or split-footed. So the feather-footed carnivores are the pinnipeds, and then the split-footed are the fissipeds. So the split-footed forms would be those that have individual toes rather than all of the digits embedded within the flipper. Now, what we can say about carnivores is that all carnivores, and I mean carnivora, have well-developed canines. Having canines does not make you a carnivore. There are some primates that have well-developed canines, and there are some South American deer that have well-developed canines as well. Musk deer have pretty impressive canines. But the other key characteristic is the fact that all carnivores have carnassial teeth. And those are blade-like or scissors-like teeth, and they are formed by the fourth premolar in the upper jaw and then the first molar in the lower jaw. So those carnassial teeth are designed to shear through muscle and soft tissue. And in the case of hyenas, they're used to slice through bone as well. Now, P4 and M1 weren't always used to form the carnassial teeth. In the fossil record, we can see that the teeth used in the formation of the carnassials were different, but currently all extant carnivores use P4, upper P4 and lower M1 for their carnassial teeth. Here you can see a diagram of what happens. Let's look at some of these animals. So the top one is a lion, and C stands for the canine, P stands for premolars, and then M stands for molar. And you'll notice these are all upper jaws, so it's the fourth premolar that forms the upper carnassial tooth. And notice for the lion that M1 is almost a vestigial tooth. There's essentially nothing left. And if you think about how cats eat, they don't chew their food, right? They slice, dice, and swallow. My wife likes cats, and years ago we had a cat, and you would go to the grocery store and buy a nice piece of liver or something like that as a special treat for the cat and give it to the cat, and the cat just puts it in its mouth, slices it, and swallows it whole, and finishes the liver off in short order. And you're flustered because you'd like the animal to enjoy it a little bit. Well, they can't enjoy it. They don't have the ability to grind the food up. They can simply cut it into smaller chunks and then swallow it whole. Well, if you're eating primarily muscle tissue or intestines or guts or something like that, that makes perfect sense. There's no need to chew it. All of that material is relatively easy to digest. Plant material that has a lot of cellulose is much more difficult to digest, and as a result, animals that eat predominantly plant material are going to have more extensive molars that are used to grind or crush. So next, look at the hyena, and the hyena probably has the largest and most impressive premolars. Note also that the first molar is almost non-existent. By the time you get to a dog, you do have M1 and M2, and the first, or that P4, the carnassial, is still rather impressive. Same is true for a marten. Same is true for a mongoose. But now look at the bear, and you'll notice in the bear, P4 is still the carnassial tooth, but it's just one small tooth in a much bigger tooth row, right? So in bears, M1 and M2 are relatively large, and that makes sense if you think about what bears are doing. Bears are eating, certainly they eat the occasional tourist, the backpacker. They'll break into your food storage, your cooler or whatever, and take all your eggs and your chicken or whatever, your hot dogs or whatever you have in your cooler, right? But they will also eat a lot of berries and plant material and green vegetation and whatever they can get. So bears are not strictly carnivorous. They do have a more varied diet, and that's reflected in the fact that they have more extensive M1s and M2s. Now, as we've mentioned before, skull shape is a good indicator of what the feeding strategy of the vertebrate is, and that's certainly true for carnivores. Let's look at the structure of the musculature of carnivores and herbivores, and importantly, let's look at the position of the mandibular condyle. This is something that we already did a little bit when we were early on in the semester. We were doing our morphometrics work on the jaws and comparing jaws across a variety of vertebrates. But here in the upper diagram, you can see the carnivore skull, and in the lower diagram, you can see the herbivore skull. The first thing to look at is the position of the mandibular condyle. So you notice in the carnivore, the mandibular condyle is relatively low on the vertical ramus. So it's maybe a third of the way up, maybe 40% of the way up. It's relatively far away from the top of the cornoid process. On the herbivore, on the other hand, the mandibular condyle is relatively tall. It's pretty close to the mandibular process, or rather to the cornoid process. And if you think about it, it's the temporalis muscle which has its insertion on the cornoid process. So if you look at those vector diagrams for the carnivore up above, you see that the temporalis muscle, that vector is relatively large, and that's because the primary muscle used to close the jaw is going to be the temporalis. And by having a relatively large distance between the mandibular condyle and the top of the cornoid process, the effort arm is going to be relatively large. And because the jaw is short, the load arm is relatively small, so the animal has a high mechanical advantage. Now compare the same situation with a herbivore. You'll notice that the vector that's drawn for the temporalis muscle is relatively small. And if you go from the angular process up to the zygomatic arch, that's where the masseter muscles would be going. That distance is now relatively large. And again, what's happening here is you're increasing the distance between the mandibular condyle and the angular process instead of the mandibular condyle and the cornoid process. So in this case, it's clear that the herbivore is emphasizing the masseter muscles rather than the temporalis muscles in terms of closing the jaw. So it's a clear indication of what sort of diets these animals are using. One animal is relying on the temporalis, generating rather significant bite forces. And of course, the herbivore is emphasizing the masseter muscles, emphasizing movement to the jaw rather than closure of the jaw. And obviously what the herbivore has to do is grind up the plant material and try and break down the cellulose. So here, diagrammatically, you can look at the difference between a hyena and a rabbit. And down on the bottom, you see what's going on with the carnassials for the carnivore, and then the molars for either the rabbit or an omnivore, right? And you see right away that rabbits and omnivores and ungulates and things of that sort are crushing their food rather than slicing the food, as is the case in the carnivores. There is another important difference in the shape of the mandibular condyle, or rather the mandibular fossa. So the mandibular condyle is going to fit into its socket, and that is the mandibular fossa. And if you look at carnivores, that mandibular fossa tends to have a C shape, and it's very restrictive. So the condyle fits into this socket, and the socket doesn't allow a lot of movement. So in a carnivore, there's essentially no lateral side-to-side movement to the jaw. The lower jaw is essentially locked in place, and what that means is that the jaw can go up and down, and that's it, no lateral side-to-side movement. If you look at something like a rabbit or a mouse, it looks fundamentally different. Instead of being this nice tight C shape, it's a much more open kind of a curved space, and you can get a lot of lateral motion in that jaw. So here, for example, up above is a mustelid, like a weasel or a ferret or something of that sort. And you can see that that mandibular fossa is shaped like the letter C, and it's going to lock that mandibular condyle in place so that the only thing that this mustelid can do is use that lower jaw like a pair of scissors. And that makes sense because it's relying on its carnassial teeth to slice and dice. And scissors only work when the two blades come into contact in a very precise way. On the lower diagram, you see an illustration of a bear skull. And recall that bears have relatively small carnassial teeth, and the major teeth in the jaw are going to be the molars. And here, if you look at that mandibular condyle and the fossa, you see that it's much more open. So the bear can get a lot more lateral movement of the jaw, which means then that the bear can chew its food, grind its food, and that makes sense because bears are eating a fair bit of plant material. They are more omnivorous than are the mustelids. So there is another interesting attribute of the carnivores, and that is that they have a baculum, also referred to as the os penis. And that's sort of interesting because other mammals have a baculum as well, including the rodents and including primates too. Human primates obviously are not in that group. But the question is why have a baculum? And there is this sort of interesting feature that is sometimes associated with the existence of a baculum, and that is that many of the carnivores also have induced ovulation. So that the female is not going to ovulate unless she's copulated. And of course, there are advantages to doing that. The female is not going to waste eggs. That's good for the male. And who knows, that may be good for the female as well. Here you can see what those bacula look like. These are not drawn to scale. But red fox up at the top, raccoon is very characteristic, has this kind of hook shape on it. Least weasel has a pretty bizarre shape. Mongoose, which is an animal that's roughly the size of a raccoon, well, smaller than a raccoon, and yet has a baculum, which is much more robust. A genet, and then a lion, which has this sort of odd sort of structure. And of course, one thing that I would like to point out is that size of the os penis and size of the genitalia are really not related to the size of the animal. Now, most carnivores have anal sacs that are used for intraspecific communication. That is for territoriality or establishment of a home range or something like that. Sometimes those are used as a defense mechanism. If you've ever encountered a skunk, you've probably been well aware of that. The smell that's produced or the oils that are produced by those anal glands are pretty potent. The good news for us is that the striped skunks, Mephidus, the animal that we have here in the most common skunk that we have here in southeast Missouri, that odor is pretty benign compared to the spotted skunk, Spilogale putorius, which is that particular odor is absolutely deadly. Interestingly, though, those anal sacs are minimal in canids, in felids, and they're absolutely absent in ursids. Now, carnivores have claws. Sometimes those claws are retractable, as in cats. In dogs, the claws are not retractable. Obviously, the advantage for having retractable claws is that you can keep them nice and sharp, so that if you're going to slice something open, they will be ready to go. A dog, obviously, if a dog is doing a lot of walking or something of that sort, the claws are going to be worn down. If you've ever seen a grizzly bear and the claws on a grizzly bear, they are impressive. They are long, and it's pretty clear that you don't want to be on the receiving end of those claws. They are quite spectacular. Now, no carnivores have opposable digits. And in fact, if you look at the wrist, the central, the scaphoid, and the lunar bones are all fused. And what that means is there's no sort of lateral motion in the wrist. All the motion is in one plane. So here you can see that all of those bones are fused to form the scapho-lunar bone. And what that does is it restricts all the motion into one single plane. And that's okay, because what these animals are doing is they're ripping stuff open. They're not going to manipulate it once they've ripped it open. The objective of those front claws is to bring the prey item down or to rip it open, something to that effect. And you don't need a lot of manual dexterity in order to do that. Look at the sesamoid bone on this illustration. What happens in pandas, both lesser pandas and greater pandas, is that radial sesamoid bone is extended a little bit, so it projects out to the medial side, which is to your left here as you're looking at this. And what that does then is it forms what's referred to as the panda's thumb. So it's not really a thumb, it's just an extension off that radial sesamoid bone. And that then enables the animal to split or to strip the leaves off the bamboo shoots. And it improves efficiency for an animal that needs all the improvements in efficiency it can get. So here you can see what the difference is between a human hand and a carnivore hand. So you see what the fusion does is it limits the range of motion in that hand as compared to the human hand, which we can wave and manipulate our hand in all sorts of different ways. Of course, that's important for us. It's not at all important for the carnivore. The clavicle is reduced or absent in carnivores. And what that means is that the limb now extends from the tip of the toes up to the top of the scapula. So the clavicle locks in the distal part of the scapula in us. So our limb basically goes from the shoulder to the fingertips. But in these animals, it goes from the fingertips all the way up to the top of the scapula. So what that means effectively is that the animal has a longer appendage, and that means then that it can have, or it has a longer stride length. And of course, there are two ways to run faster. You can increase your stride rate, or you can increase your stride length. So that simple little evolutionary change has been significant in terms of increasing the speed of carnivores. Note also that, of course, ungulates do exactly the same thing, right? And again, it's all about increasing speed. Now, when you look at carnivores, some are digitigrade. That means they walk on their toes. Those would be the felids and the canids. Others are plantigrade. That is, they walk on their palm or the sole of their foot, right? So bears and raccoons are walking on the soles of their feet, on the palms of their hands, while dogs and cats are running on the tips of their fingers. And of course, what that means then too, if you're running on the tips of your fingers, you have a longer limb and therefore increased stride length and increased speed. If you're running on the palms of your feet or so, and on the palms of your hand, then you have a shorter appendage and have reduced speed. So the size range of carnivores is pretty dramatic. The least weasel, Mustela nivalis, weighs 70 grams, so that's smaller than most of the rats that you'll find in St. Louis or New York or Chicago. Ursus arctos weighs 800 kilograms. So polar bears are up there at 800 kilos, and then least weasels are only 70 grams. That's pretty significant, right? Size is important, right? Size matters, and we've talked about that throughout the semester, so you can just ponder that for yourself. There is another important component of all of this, and that is that most of these guys have a high protein diet. And what that means then is that they have relatively short GI tracts. So the length of the intestines is significantly reduced. Well, you don't need a lot of intestines if you're eating protein. It's fiber and things of that sort that require longer intestines. Horses Cows, things of that sort, have really long intestines because they're trying to extract nutrients from a poor quality food, namely from cellulose or from fiber. Carnivores have short GI tracts, short gut retention times, and carnivores are like us in that they have a reduced cecum, so they'll have an appendix, but it's a very tiny little thing. It's not the sort of appendix that you'll find in something like a sloth or a koala bear or something like that, in which case the cecum is going to be extremely large.